Hi everyone, welcome to our weekly video, and this one is going to be covering mitosis, which is the first installment of our reproduction series. The first is on mitosis, the second will be on meiosis, which is sexual reproduction. But before we get into the nitty-gritties of sexual reproduction, we've got to cover regular cellular reproduction. This is a process by which body cells can make new cells. Now, that can be for different reasons. All right, as I jump down a little bit, growth, you want to get bigger? Well, you need mitosis. You want to fix an injury? You need to be able to repair. You need to do mitosis. If you need to replace old, worn out cells, red blood cells, bone cells, skin cells, you need mitosis. And the way this happens is by the nucleus of your cell copying all its genetic material and dividing to produce two identical new nuclei. When that cell divides, cytokinesis occurs. That division, that word, cyto meaning or referring to cytoplasm, kinesis referring to kinetic or movement. Cytokinesis is the division of that cytoplasm which occurs after mitosis has finished. Now, so if we're going to review the whole process, talking about humans, humans have 46 chromosomes. This is where the process gets a little muddy. So I'm going to use this time to actually do some drawing. I'd like you to pause and take a moment, go get a sheet of plain paper, and if you have some colored pencils, great, we can use them. Otherwise, you can just draw along with me so you can keep track of what's happening. So here's what we do. Chromosomes carry genetic material. It's in the form of DNA. Each cell has an original and its copy. And I'm going to switch over real quickly to show you the scope or size that we see here. So you know that DNA is this twisted ladder shape. All right, we normally draw it showing that twist like that. Right, well, humans have 23 chromosomes, which we label N for one full set. You get 23 from mom, so that means you're going to have 23 pieces of DNA. Each one of these would be one piece. You'd have 23 of them inside of the nucleus of your mom, and you'd get 23 from a nucleus provided by your dad. So there are going to be 23 matching types. So right here, there might be a gene for eye color. On the matching chromosome over here, there might be a gene for eye color. Now, I'm totally skipping the sexual reproduction part of that because that's for a later discussion, right? But your cells have 46, and that would be called 2N. N is just a variable like X is in math. 2N is the variable that we use to mean it has two full sets of genetic material. So you have 46 chromosomes. It makes it really hard to track that inside of the nucleus of a cell. So what I'm going to do is I'm going to change it up. I'm going to say, all right, let's talk about a muntjac. You probably don't even know what a muntjac is, but I'll show you real quick. This is a muntjac. Muntjacs are a deer or a relative of the deer that is found in Southeast Asia. It actually only has 8 chromosomes, which makes it a little easier to talk about because it only has 8 pieces of DNA that make it work. So if we pretend for a minute that we're just dealing with an organism like a muntjac, right? I have 6 colors here. Each one of these would be represented by a piece of DNA of a particular color. Now I've got blues, I've got greens, and I've got reds. I've got 2 blues, 2 greens, and 2 reds. 1 blue comes from mom, 1 blue comes from dad. Mom, dad. This would be a piece of DNA that's coiled up. That DNA takes the shape of a chromosome. It's coiled up really, really tightly. All right? And we copy that genetic material during mitosis to make what you recognize as a chromosome. It's the original and its copy. So I would be doing that with each chromosome to make each one of these an X shape. It's still represented by, in this case, 6 chromosomes. It's just each of the original chromosomes has been duplicated. So I have an original and its copy. Now, mitosis is the process of sorting these out and separating each of the copies. On your paper, we're going to sort and separate each of these chromosomes. So in a regular nucleus, all that DNA is copied. That's going to give me a broken colored pencil. 1, 2, 3, 4, 5, 6. Wow, how many broken colored pencil tips can I possibly make? 7. Whoops. How about, oops, I got too many now. I went overboard. So that's our starting point with 6 chromosomes. Well, I need to separate those into the different cells that I want to produce. We do this with mitosis. Interphase copies all the DNA. Mitosis separates it. So the first part of this, there's only 4 phases, P, M, A, T. The first phase of this is the process called prophase. Prophase is when chromosomes become visible. Then, during prophase, you have centrioles that migrate towards the poles, and they'll have some specialized protein fibers that will attach to these chromosomes. And they'll move to the next phase, which is metaphase. So this is a gradual process of the centrioles moving towards the poles until they get there. All right. Those centrioles are going to move from a location on the side somewhere. All right. I'm just picking a spot, and they're going to migrate towards the opposite sides. And in doing so, they're going to pull using some proteins called motor proteins and dynin. Each of them is going to line up near the center of the chromosome. In no particular order. I'm being deliberate in putting them mixed up because they don't form any pairings or anything like that. They just line up. You've got the mom copy and the dad copy. What did I forget? Let's see. I got my greens. Oh, I got my last red one. All right. So they just line up along what we refer to as the metaphase plate. It's not really, it's not really a structure. It's more like a location. All right. They're just going to pile up here. Now, again, in humans, sorry about that. In humans, there would be 46 of these. That's just way too many to keep track of right now. So we're going to just look at these 6 and show that once this metaphase lineup at the middle, once this lineup occurs, they're going to start to separate. Now, I drew my metaphase plate going across the middle, all right, horizontally. It's important to know no matter which way they're drawn, they will always separate perpendicular to the metaphase plate. So in my image, they're going to be going up and down. But just to make it so you understand that this can vary, I'm going to show you the next phase. As they start to pull apart, actually I should do it this way. Sorry about that. Make that a little bigger. They're going to pull apart from that metaphase plate. Even if that metaphase plate goes in a different direction, they still go towards the poles. 1, 2, 3, 4. I'm missing my last green one. If the poles are here, the metaphase plate, they will always separate perpendicular. So it doesn't matter which direction you're going, it's always perpendicular to the metaphase plate. All right? So this is anaphase. Anaphase, Ana meaning apart, right, or to separate. We like to use the memory clue away, anaphase away, right? They're going to be moving away from the center line and towards the poles. And this is just a chromosome that has two halves that have been separated. So that chromosome is pulled apart, and these are called daughter chromatids. All right, so a chromosome is made up of two daughter chromatids. When they separate, they get to become chromosomes because now they're independent. All right, they're just, hey, this is a full copy, and this is the exact replica. They're the same, and they started out this way. They just, now they're not connected to their copies anymore. So in my last pair of pictures, we're showing how now if my poles are here, my metaphase plate would have gone across this direction. Again, it doesn't matter which direction it's going. They always separate perpendicular to the metaphase plate. All right. This would be the last phase, telophase, and we're going to include cytokinesis. So telophase is going to be the moment when these separating chromatids have all made it to the opposite side, and a new nucleus is going to start to form. This is one of the characteristics of telophase. So I'm going to have one copy of each one of these chromosomes. These are the daughter chromosomes that all move to the sides. 1, 2, 3, 4, 5, 6. One more. Yep, there. And then I'd have the same over here. 1, 2, 3, 4, 5, and 6. Notice there's one of each color. There's one from mom and one from dad. So I've got a version of each chromosome from mom and dad in the new cell. That's telophase. Cytokinesis is this pinching in of the cytoplasm, forming cleavage furrows. This is called the cleavage furrow. And that will continue to push inward until it meets, and the cell will go back into interphase, and all those chromosomes will have unwound, and you'll have all those pieces of DNA mixed together doing their job. Every chromosome is in there, but now we don't call them chromosomes anymore. It's just chromatin. So the nucleus and the nucleolus may reform. All right, and so this allows the cell to continue to do its cellular jobs, and when it needs to divide again, it will. So now let's look at the second page of your notes, and we'll be able to go through this pretty quickly. This is going to describe the cell cycle. This is the series of events that the cells go through as they grow and divide. There are two main phases that we've identified: interphase, and then there's the mitosis phase. Interphase is actually divided into three sections: G1, S, and G2. I've added a couple of additional notes in here to help us with this, but the majority of the cell's time is spent in interphase. There are specialized proteins called cyclins that tell the cell when to enter each of these phases. And so a cell will go through these parts of interphase in this graphic. 3/4 of the cell time is spent in interphase. In reality, it's likely to be more than 90%. So interphase is G1, growth happens for the cell. Then it goes into the S phase. S is for synthesis of DNA. So that's the synthesis phase. And then it goes into a second growth and preparation phase where additional organelles are made and the centrioles are going to be replicated. That's the cell cycle. And then once all the prep has been done, it'll enter mitosis. And remember, mitosis is strictly the PMAT or PMAT: prophase, metaphase, anaphase, telophase. So here are a list of different characteristics of each of these phases that we just drew. Interphase is, they like to say it's the resting stage, but it is certainly busy. The cell spends the majority of its time here. Cell growth occurs here. And during the S phase, and this is important to note, that if I can draw, I don't know if I'm going to be able to draw it. I can do it. It's so tiny right now. All right. The S phase is the phase where chromosomes are replicating. Once all the prep has been done, then you can begin mitosis. That's right here. Prophase. Prophase is the time when centrioles move to the opposite poles. You'll get this football-shaped spindle forming. The spindle fibers go from the centrioles down to and connect to the chromosomes. The chromosomes will begin to move towards the equator because those centrioles and the spindle fibers are pulling them. The nuclear, sorry about that. The nuclear membrane is going to break down. There are enzymes that will break down that nuclear membrane, and the nucleolus will disappear because it's not going to be used. As they move, they will eventually make it to metaphase. That's when they line up single file right along the middle. That's right here. All right, so metaphase middle, the centromeres, which are holding the two identical copies together, they're going to line up along the metaphase plate. Once it starts to move and separate from there, it's no longer metaphase. That's going to lead us to our next phase, anaphase. Anaphase is the away phase. You can see it in this graphic here. This is an actual microscope image. You can see that the chromosomes, or the daughter chromatids, are moving away from the middle towards the poles. That's anaphase. Now, early anaphase is when they've just started to move, but late anaphase would be when they've gotten much closer towards the poles. It's just a gradual thing. So early would be just starting, and late would be almost finished. Which brings me to telophase. And telophase is when you're going to see the spindle fibers start to break down. Again, those are made out of proteins. We can build and break them down super fast. All right, the spindle's going to break down. The nuclear membrane is going to be rebuilt. And you're going to see that two new cells are going to begin to form as the cleavage furrow starts to push inward. That's cytokinesis. Remember that telophase and cytokinesis are technically two different components of the process. Telophase is what's happening to the chromosomes. Cytokinesis is what's happening to the cytoplasm and the rest of the cell. So there are some unique differences that we need to identify between plants and animals in mitosis. Plants, because they have this rigid cell wall, they can't pinch in. So they form what's known as a cell plate, and that forms across the middle instead of cytokinesis. Plant cells also don't have centrioles. The spindle fibers actually have the ability to attach to different proteins that are embedded in the cell wall. So they don't have to attach to the centrioles, they attach to proteins embedded in the cell wall. Cancer. Cancer is uncontrolled cell division. You might be thinking that's a huge jump to go from cell division to cancer, but it's not. Uncontrolled cell division is when the cyclins that the cell has are not functioning properly, and there's an irregular, shortened interphase where the cell skips this long G1 and it goes right into replication and division again. Carcinogens can cause this. There's a list of these on the electronic copy. You can access this through the module. Carcinogens can cause cancer. So can, I will ask you to add to this list here, if I can get it to click for me. How about it? There we go. Ultraviolet radiation. Well, I should say any radiation. But UV radiation is one that causes skin cancer. All right, because it could be X-rays or gamma rays. We can also add on here viruses. Also have the ability to mutate our DNA. We call these oncoviruses. They can cause cancer as well. These don't have to be bold, but during mitosis, cells contain a protein that tells them when to divide. I mentioned these earlier. They're called cyclins. I believe they still read this in English, The Curious Life or The Incredible Life of Henrietta Lacks. Henrietta Lacks is the person whose cells were collected that are used for lots of our cellular research because they divide indefinitely and they never age. They're actually, there it is, The Immortal Life of Henrietta Lacks. Because they do not die, they just reproduce and reproduce indefinitely. Cyclins regulate the timing of the cell cycle, and these HeLa cells, as they're called in cellular research, they divide indefinitely and their cell cycle is not regulated. If the cell cycle isn't turned off, cell growth can lead to the development of tumors. This leads me to apoptosis. Apoptosis is when some cells that are not destined to survive go through programmed cell death. Apoptosis is this programmed cell death. For example, when your fingers and toes are developing in the uterus, tissue between your fingers and toes will go through apoptosis that removes the webbing that actually develops there, so you don't end up with paddles or fins for hands and feet. Certain plants will lose their leaves during autumn, and that's caused by apoptosis as well. And your body has the ability to identify cancerous cells and tell that cell to really kill itself through programmed apoptosis so that it doesn't spread the cancer that it has. That's done with a p53 gene that's supposed to do that. There are a couple ways that asexual reproduction can be utilized by the different organisms in the plant and animal kingdom. This is asexual reproduction on a bigger scale, not just on the cellular level, but on the organism level. So new organisms can develop through asexual reproduction as well. Remember, this means without sex. So bacteria do binary fission, which is just taking that single cell, dividing it just through mitosis, producing two new identical organisms, and there's no change in the genetic material generally during that type of binary fission. This occurs in bacteria, protozoa, and algae. Plants and some small animals can do what's called budding. This is similar to binary fission, except that the division of the cytoplasm is unequal. Yeast does this. It'll form a little buddy. I always make jokes about that when I'm teaching it. These colonies will develop, and a hydra will produce this little mini-me that grows off the side of it. It's a bud. It's genetically identical, and that can pop off and form a new organism. In addition to that, spores, like mold spores, they produce these specialized cells that are genetically identical, and those mold spores can be carried away from the original organism and develop into a new organism that is genetically the same. So mold spores are formed because the mold has the need to spread and grow genetically the same as its parent. Regeneration is one way that mitosis is used to replace lost structures. Starfish that lose a limb can grow it back. You probably heard of cutting planaria to have them grow things back. Earthworms can be cut in half. A lot of invertebrates, meaning they don't have backbones, have the ability to regenerate. But higher organisms have lost a lot of the regenerative abilities that they used to have. We don't have the ability to regenerate a limb, but we can regenerate skin cells. Other plants will do vegetative propagation. Vegetative propagation is using roots, stems, or leaves to grow a new organism. We do this with garlic. You can take a garlic clove and you can plant it and grow a whole new garlic. We can do this with cuttings from different plants. So you can cut them and you can graft them together. I talked about this with grafting apple trees. Whoops, sorry about that. Grafting apple trees or seedless oranges. They're all those trees and plants are created using an original that they just took a cutting from and combined it with rootstock to grow another plant. Potatoes work this way. Also, they grow underground, and each potato is genetically the same. So all of these different strategies that plants have to propagate themselves, propagate means to spread, are just there to create genetically the same organism. All right, any questions about this, please feel free to contact your teacher, and make sure you get your homework assignments done and keep up with your workload. Have a great day.